welcome Dr. Sangeeta Choudhury. She is the Senior Consultant Scientist in Department of Biotechnology and Research at Sir Gangaram Hospital. Today, she is going to talk on exploring the potential of mesenchymal stem cells to target the pancreatic cancer stem cells. Thank you.
Thank you for the introduction, and I heartily thank the organizer, S Gopinath, for giving this opportunity. So this topic has not been mesenchymal stem cell. It's actually in a lot of areas in clinical trial, both having pros and cons about it. So how did we come across this particular topic actually? So a couple of years ago, one of my students in fact, two of my students who joined me about 4 years ago. And I told them I would like to, I've been working on pancreatic cancer, so I told them that they could choose the topic and they could bring forward and discuss what it would be. So they looked into the literature and came across, as everyone has been talking today, that's Neha, she's a DSP Inspire Fellow, and Poonam, as a Senior Research Fellow at ICMR. So they looked at various things in the pancreatic cancer literature, which we have been talking since morning, so I will not go into it. Yes, it's just got a 9% 5-year survival. It's drug discoveries after 2015 with gemcitabine, paclitaxel, and FOLFIRINOX, yes, but yet this combination of gem with paclitaxel is the best to date. They looked again into the literature with immune therapy, and all of them are either closed or in trial, but nothing true clinical benefit to date. And so, where do we start at it? So this is just telling you that PD-1, PD-L1 check inhibitors with the DDMR, only 0.8% of the patients are there, and so it's dismissal. So there is a need to look into a therapeutic, apart from probably the chemo or radiation. Could we look at a cell therapy? So we talked about it, and what should we be the target for cell therapy? Yes, the desmoplastic TME definitely, but also the pancreatic cancer stem cells. And so they came out with a rationale that if we could look at mesenchymal stem cells that are adult pluripotent stem cells, which could be derived from many sources, and if we could ask questions, what would be their therapeutic effect to deal with pancreatic cancer? So what is mesenchymal stem cells? These are non-hematopoietic, definitely. They are multipotent, prevalent in many sources, and what we are using is the umbilical cord. And from the umbilical cord, we dissect the bottle jelly, and from the bottle jelly, we derive the mesenchymal stem cells. So as per ICCT, that is International Society for Therapy, they have set up certain criteria to recognize MSCs, and we adhere to all these of them. They have very strong immunomodulatory effect, capability to migrate to inflammatory and tumor sites, and relatively they are immune evasive. And why are they immune evasive? So the question that we looked into it is, does it elicit an immunological reaction? Because it's a biological material. And it says no, because both MHC class 2 and HLA-G are adversely lacking MHC and increase of HLA-G. That leads to non-immunological reaction. Does it migrate? Yes. Being stem cell, it has a property. Can it be retained in circulation? Because if you're giving a drug, you need to say the half-life cannot be short. In fact, it expresses the CD47 SERT alpha, which is like eat me not. So it's not phagocyte. And so probably it can be retained. Multiple trials ongoing on MSCs, enhanced over last 10 years. So we characterized, as I said, and then what we went ahead to do it is, we did a proteomic analysis using the LCMS non-targeted to look into what all, it might be having. What does it have? Why should we use it? So we also differentiated that's one thing that the MSCs can do is they can have lineage differentiation, especially adipogenesis, osteogenesis, and chondrogenesis. So this has to be one of the property, otherwise we haven't actually kind of cultivated proper MSCs. So they do express certain genes, that is 105, 73, and 90. They should be absent for expression of 34, 14, any lineages for that matter. Now what we came across is FGF2, vinculin, and CXF were very highly abundant. And probably this could be, the reason for transversing, that is from epithelial to mesenchymal. They play a major role. Of course, they did have IL-4, IL-12, and these are the anti-apoptotic cytokines, and so that means they were expressing even this. So this made us think that it could be used. What we also went ahead and did is, we again did a proteomic analysis of the secretome, and what we find is, of course, glycolysis, TCA, JAK-STAT signaling, and hedgehog signaling were being highly molecules expressed by them. And so we went ahead, and this is just the protein expression, the secretome analysis, which I will go ahead. When the students were looking into it, they found as early as 2002, Amelia Bartolomeo, she was the first one to say that MSCs could modulate immune response. And she worked on baboons, 6 baboons, where she showed, so that is when it's allogenic, because we are looking at allogenic and we're not looking at autologous. So this gave encouragement that a hypothesis could be derived and could work towards it. So this is the outline of the experiments that they went ahead to do. They grew the pancreatic stem cells in spheroid format, and thereafter they characterized it, validated it, and did it. So now, as we've been talking about heterogeneity in pancreatic cancer, along with its mutational analysis, what you find is that 2 different cell lines, morphologically, proteomically, and even later on in genotype, it showed that they differed very different from each other. And so, this is the distribution of their, stem cell markers, and the right side corner on the top out there shows that CRC and CD44, CD24 combination would be the best to recognize a panc-CSCs. And we did go ahead and show that all the panc embryogenic cells, genes were being expressed by them. This is the dose response to these, to look into the pancreatic stem cells and pancreatic tumors. And yes, after passaging for at least 12 passages of the spheroids, we use these models to go ahead, and it does show clearly that by LTT, the gemcitabine as well as 5-FU, where you can see very high doses could give the IC50 for that. Migration capability was similarly being affected by if you are going to use. So here it is, we're talking about how under inflammatory conditions, these are, they have different phenotypic and migratory properties. So we say the capability between the migration between the two significantly different, and they had differential capabilities on this. The cell line itself goes ahead to do it. This I've already told you that we did that. So what came out of the result on the pancreatic proteomic? The 2 cell lines actually are probably being regulated by 2 different pathways. The MIA-PACA, that is the G12C, is via the 2GF SMART pathway, and PAN-C1 with the G20 is via the PI3. So if you take a pancreatic cancer patient, be it stage 2, 3, and you give a general which probably that's one of the major failures, that you need to personalize, do their genomic, and then probably you will get a better idea. How can we target these? So these are our papers only saying the potential of stem cells, and we also did, that's another story where we used synergistically vitamin D with MSCs, which showed good results. So what is when we treated the MSCs with the tumor cells, one very interesting figure came out is that PD-L1 being expressed by them. And so that is a very big step to say ahead, that probably we could use the nivolumab and the rest of them being used, which is till date shown to be not effective or very negligible effect. If we can activate them, probably that would be one way of, some patients might benefit from such a thing. We went ahead and looked into the modulate functional capabilities of them, like migration capability, tumorigenic markers like MUC1 and CXCR4, and also angiogenic markers CD31. And as you can see out here, that tumor cells showed compromised migratory capability in presence of and treated with WJMSCs. Glycoprotein MUC1 and CXCR were downregulated, as well as CD31 was also shown to be reduced expression. Then we said, what about the stem cells? Because the stem cells cannot be living on its own. It's a give and take between its microenvironment and the cell. If you segregate the cells and grow them, and every time you deplete the media that they have been growing, you will see that these cells grow much slower and delayed response. So we wanted to look how these two together works about it. We looked into the apoptotic markers, we did the qPCR, finding out the apoptotic markers like caspase 3, 9, BAX, and also we then went ahead and looked where was it affecting in apoptotic markers. And what one comes out in is this, that they are, as at both phase, early and late phase, the secretome can be effective in regulating the cell cycle. So this is the wound healing, because we are using the MSCs, which are known to be, till date, known very well in cosmetic area, where they heal the wound. Even burn patients are given stem cell treatment. And so we looked into the wound healing, that is whether they can overcome the treatment, both the secretome as well as in comparison with gemcitabine. And one can see that they had reduced invasive capabilities, which were reduced by treating with secretome, in compared to gemcitabine that you can see. Flow cytometry data again showed that FCAM, CXF, and MUC1 were downregulated. And these are the different pathways that we went ahead to explore. What could be the pathway? Do they follow one? Do they have multiple actions? Because we know that cancer doesn't follow one single pathway. And so we looked into not only just EMT and hedgehog, but mTOR and GSK also. And what we feel is by the data, there were other data that I have shown out here, is the secretome appears to be regulated by the MUC1 STAT3. And otherwise, it also seems to be there's some effect on the EMT, but only on Z2 environment, not on TWEST or other markers of EMT. So this is what comes out from the student's hypothesis, that could we say it is the next-gen treatment option? Because what appears over here is WJMSC, when it's treating, the model is that it is the GSK3 beta could degrade the MUC1 beta-catenin complex and thus cause the apoptosis. On the other hand, if the G12C or G12D is allowed to go on its own, we could probably target the AKT mTOR or the SMART PI3 pathway. And so what could be lying in future? Actually, there is another two students following up on this, where we have patented 2 exosomes derived from mesenchymal stem cells, and that's what I'm not presenting out here. And what we have done is we have engineered the exosome to express trait, incorporated into it an anti-mTOR, and on the other hand, another model is doing an siRNA for glycopan, AKT, and KRAS G20. And all the 3 of them together, so we have prepared our model using the mammoprint, because we know we are in a hospital and we don't have access to animal. Well, anybody can, I'm available to collaborate with anybody who can help us out in getting the animal model for KRAS, which is most probably we would require as a G12D model. And that's difficult to come by in India, so we thought about going ahead with So that's the end of the story. This is the entire Department of Research, headed by Dr. N.K. Ganguly, who was the ex-DG ICMR, sitting in the center. We're very proud to have him with us. And I have fundings supporting me from ICMR in Australia as well as the DBT. My clinical teams on this, giving us certain patient sample and the tissues, is Padma Shri Dr. Samira Nandi, both of them are from GI surgery, and also Dr. Soumitra Rawat. Our clinical team, the students are enrolled with Jamia Hamdard and TU, so we have our collaborators out there. Expert teams, I work in collaboration with Rutgers for many of these models, so they are always there to help us out in our this. Thank you so much.